If you can all find your seats, we'll get started with session 3. Now, despite what the slide says, I'm not Ken Anderson, but be patient. Ken will be here in just a few minutes. He's finding his way to the front. But I wanted to give a few introductory remarks. How did you get the mic? Go on. Now it is. Okay. So, although the slide's up there, I'm not Ken Anderson, but he will be coming up here in a few minutes. In the first few minutes, what I'd like to do is really give you some introductory remarks for this session, which is flexible drug development and decision-making, accommodating new insights. Because what you've heard from this morning's session was that along the course of a drug's development, even before it enters the clinic, throughout its clinical development life cycle, and then afterwards in what we call life cycle management, we're always learning something. That could be something about that particular compound. It could be something about that pathway, about the target, about that disease. And the question that we're being asked in this session is, how do you incorporate that knowledge into your thinking, your strategy? Remember, it could come from any one of a number of places, but what we do know is that it's coming at us very fast. It can come from insights into the molecular biology or the genomics of cancer. It can come from some companion diagnostics or imaging, non-invasive imaging, which is really moving at an extraordinary pace and now becoming much more specific to certain diseases. It could be coming from clinical real-time monitoring of patients, so serial liquid biopsies, if you will, that allows us some insight into what's going on in the patient even before it's clinically apparent. So the question here is, how can we integrate that into clinical development and do that in real time when it has meaning? Now, we've heard about the evolution of clinical trials from phase 1, 2, and 3 to a more continuous approach. And that makes it even more difficult because when you have a drug that's moving quickly and then you have a new insight, potentially into selecting a group of patients who are more likely to benefit, when do you know that you have enough information to make that decision? If you see it from a preclinical setting, are you certain enough that that's going to predict what happens in the clinic for you to change your clinical development strategy and limit the development to that group of patients? Or do you open up a separate cohort? Or do you wait until there's more evidence? How do you know that the tool that you have is reliable, is going to be reproducible, that this is the right one to use as opposed to an entirely different platform? Because these questions matter. If you make the wrong decision, that can not only delay you, it could actually derail you. And as we all know, it's very rare that failed drugs get a second chance. So just as as Mark Retain has implored us to find the right dose and the right schedule, we have to also make the right decisions as we go along and make them at the right time. So that's really the questions that I've asked the panelists and the speakers to present, is their experience with drug development from very different and complementary perspectives, and how information that came along during the course of that therapy's development changed the course and allowed them to identify where the real benefit was from that. So I'm delighted that our first speaker is going to be Ken Anderson from Dana-Farber, where he's the Kraft Professor of Medicine, also head of the Myeloma Center. So Ken, please join us.
Thanks very much. So over the next few minutes, I'm going to talk about biologic activity as to its utility to inform drug development. And multiple myeloma is really the paradigm for targeting the tumor in the microenvironment. That's one of the major points I want to make. Because we can get the cells, we can get the microenvironment, and we can make laboratory and animal models quite easily that target interaction between the tumor and the host, pathways within the microenvironment or within the tumor cell, or cell surface molecules. Now, this is the track record for myeloma. We've had 4 new classes of drugs, proteasome inhibitors, IMiDs, HDAC inhibitor and monoclonal antibodies, and these have been active in the preclinical models and overcome conventional drug resistance and rapidly moved to clinical trials. There are a total of 18 now FDA approvals in the last 13 years since 2003, and people are living 3 to 4 times as long. That's shown on this picture as well, which shows that in fact, the classes of drugs and when they came along. And this is the survival on the right, and this is only up to 2010. I think it's fair to say that there are some patients now who end up having a normal lifespan, appreciating the value of some of the drugs that we've most recently had approved. So as this conference is talking about the obstacles to conventional drug development and what we're going to do to make it seamless, this particular cartoon is the traditional model where, in fact, academia, industry, and academia sort of play ping pong here or whatever. But I think one of the things that's happened in multiple myeloma is there's been a real blurring of these spheres. So in academia, there's a lot of validation that goes on. Nowadays, there's actually even prototype inhibitors that are made. And so there is, in fact, a movement on the early side, much more towards the bedside, and many bench to bedside and back examples I want to show you. This is really what's happened in myeloma. I think we actually try always to put patients first, and the team members represented in this room are academia, biotech, pharma, the NIH and NCI, the FDA, which I want to give a shout out to for having been so proactively on the side of patients, and then the advocacy groups who really have been very instrumental in this progress. So the first drug, the immunomodulatory drug thalidomide, was used empirically, as many of you know, because it was an anti-angiogenic agent at the behest of a patient's spouse. Laboratory work in our lab and otherwise showed that this drug, this class of drugs, work in the microenvironment, targeting the tumor, blocking adhesion, inhibiting angiogenesis, but most importantly, stimulating the immune response in the host against their cancer. It wasn't for years till we actually knew how these drugs were working. And in fact, this is sort of a cartoon that many of you will know. These drugs, the immunomodulatory drugs, bind to the cereblon complex and turn on the degradation of proteins. In myeloma, it's Ikaros 1 and 3, and downstream IRF4. That's very significant because now we can understand, and it turns out many of the immune effects go through the same degradation of protein. But in terms of informing based on science, which is what I'm supposed to talk about, the next generation of drug development, we have a whole new class of drugs coming to a clinic near you, which is going to exploit this idea. The idea is that you can have some medications that might be called degranomids. They look like IMiDs, or maybe they don't. But what they do is they bind to cereblon, and they have a linker so that you can degrade the protein that you want. So you bind to cereblon, the ubiquitin tag is put on a protein, whether it's cell surface, cytosolic, or otherwise, and actually degrade the protein. But that grew out of knowledge of how the IMiDs actually worked. Now, the other class of drugs, as you know, are the proteasome inhibitors, and you know that myeloma is the hallmark, the actual form fruste of secreting excess monoclonal protein. So blocking the degradation in the proteasome was, in fact, the basis for the use of bortezomib initially, and now a subsequent generation of second-generation proteasome inhibitors. But in the context of what I'm saying, the tumor and the microenvironment, which I think more and more we need to think about in the days of immune therapies, this is a cartoon that just tells you that bortezomib here acts on the proteasome in the cancer cell. But I'll just point out in the middle center here that in patients, as in our models, it has other activities. It acts on bone disease, for example, to inhibit osteoclastogenesis and promote new bone formation. So this concept of science making models and understanding that activity not only in the tumor itself but in the microenvironment, I believe to be very important. Having shown that, in fact, blocking protein degradation was important in the proteasome, We and others went on to block protein degradation in this alternative pathway, the agrisome pathway, using HDAC inhibitors. And in fact, if you block both pathways, the proteasome here and the agrisome here, with proteasome inhibitors and HDAC inhibitors, this precipitated or derived, if you will, a phase 3 clinical trial which went forward with both inhibitors, and the first HDAC inhibitor was approved earlier this year. We in myeloma have been late on the monoclonal antibody front, but we have been looking, some of us, for almost 40 years for these things. But I'm proud to tell you that, in fact, we have a couple of targets on the cell surface that I think are important that I want to mention for two reasons. They're antibodies that act, as all of you know, by ADCC, CDC, or blocking agonistic or triggering death pathways. But they represent the combination approach that can be modeled in preclinical models and translated to the bedside. So the first one that you all know is elotuzumab, or Implicity is the other name. This actually targets SLAMF7 on myeloma cells, which is universally expressed. Lots of biology in our and other laboratories went forward, and it did derive a single-agent clinical trial in relapse disease, which achieved stability of relapse refractory myeloma. That ordinarily would have been the end of the drug development story, but preclinical data showed that the immunomodulatory drug lenalidomide added to this and other antibodies increased ADCC. A phase 2 result of the combination had a large number of responses, 92%, and a phase 3 clinical trial showed a 5-month progression-free survival advantage when you added lenalidomide to elotuzumab, so the combination was approved. Daratumumab is another lesson in terms of drug development. I actually found the antigen called CD38, back then it was called T10, in 1983. That would be 33 years ago. And why did it take so long to get to the clinic? The reason it did is because we were thinking about science, and we said this antigen is on all immune cells that are activated. It's on all blood cells. It's on half of the hematopoietic progenitor cells. Even worse, it's on endothelial cells. We couldn't possibly have a therapeutic index or window, but suffice it to say, as is common for me, I was wrong. And so this antigen, CD38, is expressed on regulatory T cells, and at least one way that it works is depleting the regulatory T cells, allowing the immune response to occur. Suffice it to say that this is another example of an antibody that combination approaches are much more efficacious. As a single agent, it worked about 30% responses, but in preclinical models with both IMiDs and the proteasome inhibitors, there was a marked increase in activity. And just within the last month, the FDA has approved daratumumab with both lenalidomide dexamethasone and with bortezomib dexamethasone, both in the New England Journal, based on a 2/3 reduction in the likelihood of progression or death. So this is not a small result, and very, very impressive. Now, in the theory of combinations, we like to put drug classes together predicated upon science. And because of short time, I'll just point out, if I can here with this pointer, this triplet right here, which is lenalidomide, bortezomib, dexamethasone, which has 100% response, 3/4 very good partial, and half complete responses, including molecular CRs. So combination approaches with different mechanisms that I showed you earlier achieve a high overall and extent of response. So good, in fact, that we're now, as a myeloma community, asking fundamental questions. So here's an example. Now, over 14 or almost 1,400 patients with new myeloma have gotten the combination of lenalidomide, bortezomib, dex. Half of them went on to get a standard transplant, which is the standard of care, and half of them over here did not get the transplant. And we've been able to look and see and measure minimal residual disease and lots of science along the way. The reason I show you this is combinations of novel agents predicated upon preclinical science have achieved unprecedented results here. This randomized trial is soon to come out in the New England Journal, but drugs only or drugs plus transplant, the transplant wins on progression-free but not overall survival. But look down here, which is the reason I'm showing it. 2/3 of patients in the drugs only and 80% in the transplant achieve minimal residual disease, which honestly heretofore we have not been able to achieve in multiple myeloma. So new agents, combinations of agents predicated upon science can achieve an extent and frequency of response and outcome that we simply haven't seen. So I'm going to just finish with a couple of comments about how at least I'm thinking about using science to go forward in terms of picking the winners and designing clinical trials. So firstly, we'll talk about what can we do more about degrading proteins, what about immune, and what about genomic abnormalities, just one example of each. Well, I already mentioned to you that we have a lot of proteasome inhibitors here. What about blocking upstream of the proteasome, the so-called deubiquitinating agents? A protein has to get ubiquitinated, then it's marked for destruction, but it has to get the ubiquitin taken off to bind to the 20S core of the proteasome to get degraded. So if you block the DUBs, or deubiquitinating agents, you're blocking upstream of the proteasome. Here's 5 patients with myeloma here, whose tumor has the nerve to be resistant to bortezomib, but it's sensitive to a DUB inhibitor. This is a cell line resistant to bortezomib sensitive. So we have a clinical trial ongoing as we speak, which is achieving responses in the context of a drug that does not inhibit the proteasome because it's acting upstream of the proteasome, does cause a massive accumulation of ubiquitinated protein. It's just upstream of the proteasome, and where it looks very early but promising for overcoming proteasome inhibitor resistance. What about checkpoint inhibitors that have been so nicely described here? I'm sorry to tell you, in multiple myeloma, pembrolizumab had no single agent activity, but we're actually quite positive about the combination approach based on, I hope, preclinical data. So here's the evil myeloma cell expressing PD-L1, the immune effector cells that you know well expressing PD-1. But what's different, and in my theme of the tumor and the microenvironment, is there are other accessory cells in the microenvironment, myeloid-derived suppressor cells and plasmacytoid dendritic cells that express these checkpoint inhibitors. So if you block here with PD-L1, for example, antibody, you take the brakes off here, but you abrogate the growth promoting for the tumor and the immunosuppressive activity of these accessory cells as well. And we can model this. I think we're very privileged to have blood cancers to study. So this is a patient, autologous tumor cell killing by autologous T cells and NK cells. There's a little bit of baseline activity, but if you add in PD-L1 in the red or PD-1 in the green, you can augment, or both in the purple. You know that there are cells in patients that are just trying to do the right thing if we would let them just release and do it. We can model by putting these accessory cells into the model system. So here's the same experiment, killing autologous tumor cells here, but these are CD4, CD8, NK T cells. This is autologous tumor, autologous effectors, but this is in the presence of autologous myeloid-derived suppressor cells that are trying to shut down the system. So we can model these things, and what I'm trying to get at is just summarized in this one slide. This is killing of autologous myeloma cells by autologous effectors with checkpoints, PD-1 and PD-L1, with lenalidomide and checkpoint inhibitor. So combination immune approaches predicated upon preclinical science. Some of you may know that there's very early exciting data that was presented at ASH for this combination, even in lenalidomide resistance. And the final science that I want to mention is just a bit about genomics. This is one of my new patients that just sat down for a new patient visit, who has 5,300 substitutions, 51 deletions and insertions, and 49 rearrangements. And when they relapse, those numbers go more than double, and they're not the same. So we have, Houston, we have a problem in myeloma. We have marked heterogeneity to start with, and there's ongoing DNA damage and evolution. So I just want to leave you with a couple of thoughts. We are trying to target certain pathways in multiple myeloma, And one that I'll just mention to you, and that was new information at ASH as well, is in the patients who have the 11-14 translocation, we, as is true in CLL and some lymphomas, are targeting BCL2-related family proteins with venetoclax. And what was presented at ASH is this 40% response with venetoclax goes up to 80% with venetoclax and bortezomib. But what I think is more likely is we need to take advantage of the biologic phenotype that results as a consequence of what I call this genetic chaos. So 2 quick examples of this. Here is an example of decreased copy number here in leukemia, lymphoma, and myeloma of something called YAP1. Not mutations, just decreased expression. So what does this mean? I said a minute ago there's all of these abnormalities. There's lots of ongoing DNA damage. There's activation of DNA damage response pathways, as shown here. But these cells don't die. Why don't they die? They should die with all of this damage. Because YAP1 here is in the pathway of P73-mediated apoptosis, and it's missing. So a clever postdoc, Francesca Cottini, in my laboratory, has actually fixed that problem. It turns out there's a kinase that is suppressing YAP1. With a nanomolar inhibitor now, you can restore YAP1 expression and get death of these cells that are programmed because of all this damage to die. They just have the fuse put back in to the death pathway. And then the final thing, and this is brand new, is if we can't target with multiple drugs, could we use one targeted agent to target the microenvironment? And I think that's coming in the future, not only in myeloma, but in other cancers. So this is KDM3A, a demethylase, which is active, overactive in myeloma. It turns out because it is, there are transcription of genes like KLF2 and IRF4. IRF4 is a hallmark central abnormality in myeloma. If you block KDM3, you can restore methylation on these promoters, and not only inside the myeloma cell, but you're impacting biology, homing, cell survival in the microenvironment by hitting one target and changing methylation. So what I've tried to do is to tell you that in myeloma, we've been very blessed. We have the ability, because it's a blood cancer, to do tumor cells and the microenvironment. That has allowed us to have single agent and combination be validated preclinically and rapidly moved to the bedside. The clinical trials have been informed by biomarkers, and the combinations were also defined by usually synergistic activity. This would never have happened if it weren't for the collaborative effort that is here and that is represented in this room. And I think in the future, I call these Achilles heels. Weaknesses, vulnerabilities, too much protein, target the proteasome, et cetera, et cetera. I think if we think about the biologic consequences of the genomics of cancer, that presents a whole new opportunity to develop drugs based on science. So I think I'll stop here.